I hope that this little workshop encouraged you to try some bioinformatics and analyze the single-cell RNA-seq or whatever single-cell technology you want. I first wanted to give you a little background about myself and say that I am actually 50% wet lab, 50% dry lab. I do not have a computing science background. I am a biologist, and I did my PhD in molecular biology, which was focused on epigenetics and genomics, where I had to do a lot of next generation sequencing techniques. And that's why during my PhD, I decided to learn bioinformatics in order to be able to analyze my own data. So I'm mostly self-taught, and I'm telling you this because if I was able to learn it, everyone else can, and I really encourage everyone to at least try and give it a go. And so some of the things that this is kind of my how I started. So at the beginning, I just decided to learn some basic R because everyone in my lab that did analysis was using R. And that allowed me to then be able to analyze and visualize some bulk RNA-seq and some bulk ChIP-seq. And when I felt a bit more confident on these skills, I decided that I also wanted to be able to pre-process this RNA-seq and ChIP-seq. And for that, I actually needed to learn some bash in order to be able to work on the HPC. And then I had to do some single-cell RNA-seq experiments. So I decided to learn the Seurat package, which is an R package to be able to do all of the single cell analysis. And then now I've dived into a lot of the single cell stuff, and that has my skill set has improved like I've now been doing even more things. But yeah, so you have to start somewhere, and I would say probably some basic R is always good. And if you just want to do single cell, then basic R and Seurat is the way to go. And I wanted to share this TikTok because I thought it was really funny. Because this is like what people think bioinformaticians do all day. They just write, don't make any mistakes. They're just like machines. They know exactly what they are doing. But in fact, it's really not like that. And we are figuring it out as we go. And, you know, a lot of it is Googling how to do something, looking how other people do other things. And this is basically how you spend most days, which is Googling, copy pasting, trying to figure out how to fix that error that you had. And so what I'm going to address today on the talk are basically all of the steps that you require between the FASTQ file is generated by the sequencer until you have some sort of output that you can understand and have some biological meaning. And I will go through every single step and kind of what things you have to watch out for and the different options that you have for every single step, which you will see there is a lot of options for a lot of things. This is going to be the recurring theme of the workshop. But I'm not going to show you like a hands-on of how to do something because we don't have time for 1 hour. So I will just give you some resources and some points to consider when you are starting. So for the first pre-processing step, which is from the raw files, so the FASTQ files, to a count matrix. So this is, for this step, you first need to align the reads that are in the FASTQ file to your reference genome. And then once you have aligned these reads, you need to do UMI deduplication. So you need to remove any PCR duplicates that were generated during the library. And this is very easy for single-cell RNA-seq because we have the unique molecular identifiers that mark each molecule that is captured per cell. So every time you see a UMI in a cell that is repeated, it means that it has been duplicated during the PCR process. And then once that aligned reads are clean, you can then quantify how many reads are present per gene and from which cell they came from. And that will then create a count matrix in which your cells are your columns and your genes are your rows. And then a bunch of numbers in that table. And this is basically the object that then you can import into R and into Seurat pipeline and do all of the rest of the analysis. Here to make things simple and for the purpose of this talk, I'm gonna mostly focus on single-cell RNA-seq, in which, as I said, we have the cells versus the genes. But there are other single-cell technologies which don't look at the genes, but look at other things. So for example, site-seq and AppSeq, you will have your cells in the columns, but instead of genes, you have the antibody-derived tags. And then for single-cell attack-seq or single-cell chip-seq, you don't have genes, you have regions in the chromatin regions as your rows. And in spatial transcriptomics, you have genes, but in the columns, instead of cells, you have spots on your slide. So obviously is, depending which technology, you will have this matrix, but for basically each step of the pipeline will be the same. It's just that there are some things that will have to be different depending on which technology you have. And there are then some specialized packages for some of these other technologies that are not single-cell RNA-seq. But yeah, I will mostly focus on single-cell RNA-seq. And then here I'm just grabbing some of the examples for processing pipelines that are available. There are probably more, but this is some of the most commonly used. And each pipeline of software, so basically each company that has a single cell technology has developed its own pipeline. So for example, for 10x, you use Cell Ranger, which you can download in your cloud, and it's pretty straightforward to use, but you do have to download it into an HPC and then run it yourself. For the Rhapsody platform, you have the 7 Bridges website option, in which is much easier. So you actually just upload your FASTQ file, and then you just click and drag your references and your parameters, and that gets sent to a cloud, and then you just get the output. And you don't need to know any bash or any anything like that. And that would be, for example, the easiest way to do it. And then there is other pipelines which are maybe a bit more flexible for other technologies, or that you can modify for different technologies. But this will they will be more hands-on, and you will require more expertise to do those things. So here again, maybe something to consider when you are deciding which platform you want to use will be also like what are the resources for the pre-processing step that you have. Okay, so once we have the data matrix, the next step that we want to do is data cleaning. And I would say this is probably the trickiest part of the single cell analysis. So some of the tools that are most commonly used are the ones I've put here. So you have Seurat which is in R, which is probably the most commonly used tool, the one I'm most familiar as well. But there is also a Python-based tool, which is called Scampi, or Scampi. And then SIGGig, which is actually is an app. You don't need to know coding, and it's more click and base, and I guess it's easier for people that are used to flow cytometry analysis. So that one is very good, but there is a slight disadvantage of that one, and is that it actually requires quite a lot of computing power. And if you have a lot of cells, because it's less efficient than just doing it on Seurat, on R directly. So if you have a lot of cells, you might not have enough power on your computer to be able to process it. But maybe for smaller data sets, it's a good way of starting to look at it. And I personally don't have a lot of experience. Okay, so the first, quality control. So we need to, once we get our matrix, we need to remove empty droplets. So these are droplets that don't have a real cell inside, and they might have some ambient RNA or some debris or some things. So for that, we use empty droplet tool, which is, I would say, is the most commonly used. So there is also another one called SUPX, but by far the one that everyone is using is empty droplets. So that I would recommend starting there. And it predicts based on the number of genes when is like the dropout, so it basically aligns all of your barcodes based on the number of genes, and more or less when there is real cells, they also usually all have the same amount of genes, and then there is suddenly some barcodes that start dropping the number of genes, and it estimates where is the cutoff to make, say, oh, these are empty drops and these are drops with cells. Then we have to also remove doublets, and that I've personally suggest doublet finder. Here you do need to put the empirical number of doublets that you expect based on the doublet rate of your technology, and then it finds the cells that are most likely doublets, like that number of cells, and then it looks at all of your cells and says, oh, these ones are the most likely doublets because they have transcriptomes that cluster in different places. You have to also remove if there is any damaged cells. So those usually you look by, those cells have high mitochondrial fraction. So this is because if a cell is damaged and the membrane is not, it has holes or is lysated or is dying, the cytoplasmic RNA might have left, but the mitochondrial DNA is protected and it stays in there. So that's why you usually have high mitochondrial fraction on damaged cells. But here you also have to be careful that, especially depending on your sample that you're looking at, because you might have a system like cancer where you have actually cells that are really high metabolic and they have high mitochondrial activity. But so usually what I do is look at cells that have high mitochondrial fraction, but also low number of genes. And that you make sure that the cells that are removing are actually damaged cells and not highly mitotic cells. And finally, if you have chose to use sample ID tags, where you have tagged different samples within the same run, you will have to do demultiplexing, which is all of your cells are coming in one run, but you have to then assign which sample was from which based on these tags. And I will explain a little bit more later. But the good thing about using that approach is that is an easier way to also find doublets, because if you have two tags present in one barcode or in one drop, it means that there was two cells in that droplet. So once we have cleaned up our data, we don't have doublets, we don't have damaged cells or empty droplets, then we can normalize the data. And the goal for normalizing the data is to remove technical variability between cells. Single cell RNA-seq is quite noisy, and it has a lot of dropouts, so some genes are not detected and have a lot of zeros, and that can create problems. So after normalization, the goal is that the variation that you have in your data should reflect the biological heterogeneity rather than differences between your cells on gene abundance or sequencing depth. And there are a few methods to do normalization. The log normalization was the most, it's kind of like the most standard, and is the one that has been used based on, because it's the one that it was mostly used on bulk RNA-seq. So that's the one that most people started using. But now there's been other methods such as SC transform that the Seurab team has developed, which is based on negative binomial regression. But basically what you need to know is that it's been performing better for single cell analysis, and it's been developed especially for single cell analysis to tackle the problems that single cell data usually have. So yes, I will recommend that one. Okay, so the next step is doing data correction or integration if it's needed. So sometimes you have only done single cell in one sample, and then that's it. You don't need to do this step. You're lucky. But if you do have several samples, especially if they have been run in different batches, you might want to pull them all together and analyze them together, and that can have problems. So this step tries to remove technical variability caused by batch, platform, or even if you have different modalities that you want to combine. And there are basically different approaches based on the problem or based on the objective of what you're trying to accomplish. So we have three types of integration. The first type of integration is horizontal integration. And in this case, the genes are your anchors. So this would be when you want to combine different captures that have been captured separately, or different batches, or samples in different lanes within the same 10x run. But obviously you have one separate FASTQ file for each of your samples and one different matrix count matrix, and then you want to combine them. And you want to make sure that if there is a batch effect or a technical variation, you remove that technical variation and you are not counting that. So yes, so it's for independent group of cells, but it's the same type of modality. And there is many different ways of integration. There is this review that I wrote here where they look at all of the types of integrations and some of the advantage or disadvantage of some of them. Seurat, the version 3 of Seurat had one type of integration, but now the new version, the version 4, has the RPCA, which is very good for batch effect correction, and is a softer integration so that you are also not overcorrecting. And you can also kind of dial down or up the amount of correction depending on how much you need. Then you have vertical integration. In this case, the cells are your anchors. So you have different modalities, but that you have collected in the same cells. So this is like a multimodal approach. So for example, you have APSIC and whole transcriptome RNA. And I would say this is the easiest way of integrating the data because you know exactly which cell is coming from, so you can really correlate very easily the gene with the antibody or with the chromatin accessibility of all of that. But you have also some ways in Seurat 4 in which you can combine the information from both using a weighted nearest neighbor, I think it's called. They combine the information for both to generate a UMAP. And then finally, we have the diagonal integration, which is by far the hardest, which is when you have different cells and different modalities. So there is no anchors here. And there's been some methods that have tried to tackle that, and I've brought some there. But for example, sometimes what people do is try to assign the region to a gene. So if you have for example, in a taxic, you have regions in the chromatin, but if you can try to assign that regions to a gene, then you can then again have the gene as an anchor and integrate using the horizontal integration. But obviously you might lose some information if there is regions that do not have a clear gene, for example, distal regulatory regions. But yeah, again, I've brought here a review where you can read more about it and try to look at what integration will be best for the problem that you have. Okay, so once the data is now normalized, corrected, integrated, it's beautiful, it's perfect. Now we need to choose the genes that you will use for the dimensional reduction, so to visualize it. So usually the feature selection or feature is in case it's not genes or whatever regions are, but you want to select the ones that are the most variable in your data set. And we usually select between 1,000 and 5,000 highly variable genes. This is obviously depending on how heterogeneous your data is or you expect it to be. You can actually use all of your genes to do the next step, but it will make the computing slower and you will be probably using a lot of computing that you don't really need. So between 1,000 and 5,000 is a good number. And you need to select the genes after you have done the data correction and you have removed the technical variability, so that the genes that you are selecting are based on the biological heterogeneity and not based on a batch effect. And then we will do the dimensional reduction, which here what we want to do is to capture the maximum complexity that we can in our data in a 2D in a 2D graph, 3D sometimes, but 2D most of the times. To reduce to do this, usually you start with a principal component analysis, which looks at which list of genes separates my cells into groups, basically. And then it looks at which other genes will now separate it based on something else. And then the next one will be like, oh, now we will have a different group of genes that separate these other two groups. And then once you have done all of the PCAs, you can see how many dimensions you need to separate my data, and when does this principal component analysis, stop separating the data at all. And then once you have selected how many dimensions you need, then you can do a further dimensional reduction that uses this PCA information, and that is the TSNA or the UMAP. And then why TSNA, why UMAP? Some people like TSNA, some people like UMAPs. So basically, a TSNA looks at what cells are the most similar to each other. But it's not very good at looking at how different a cell is from another cell. While the UMAP is better at telling, oh, this cell is quite different from this other, so we will put it farther away, but then there is this other group of cells that is even more different, so that one is going to be even farther. So basically, the distances in the UMAP are more representative of how different the things are, while the distances in the UMAP are not so good at telling how different things are, but sorry, the distances in the TSNE. But the TSNE, when things are quite similar to each other, you can still see more differences, while the UMAP, when things are very similar to each other, are all very, very close together, and it's kind of hard to see. So I would say try both. The more you visualize the data, the more you understand it, and I would say it's better to look at the data at least once in both ways, and then you decide what do you think is the best for your type of data. Okay. So the next step, once you have visualized the data and you have construct either a UMAP or a TSNE, we need to cluster it. So make groups of cells that are similar, will be all in the same clusters. And when another group of cells is different, it will be in a different cluster. And to do this, you can use the K-mean cluster. This is unsupervised, so it uses the information from all of your genes or the 2000 features that you have selected, but you do not say anything about, oh, I want this to be one cluster. It's unsupervised. And also, I want to say here that the number of clusters really depends on which resolution you use, and you can increase or decrease the resolution. So if you want more resolution, you will have even more clusters, and you can increase that until you have many, many, many different clusters that are very, very small, or sometimes even they start to kind of not be so well defined and they are starting to merge between each other. That's probably when you know that you have gone a bit too far with the resolution. But what I'm trying to say here is that because there is a cluster, because there is a cluster doesn't mean that you have a different cell type, because the number of clusters depends on how much resolution you are adding. So you might sometimes have one cell type that is two clusters together, or sometimes if your resolution is too low, you might have one cluster that actually, if you increase the resolution, will be divided in three, and when you look at this identity of those three, are actually different cell types. So this is something that you have to play around and look at it. So, and the number of clusters will also depend how many cells you have. Sometimes when you have many, many cells, it's hard you can increase the resolution a lot, but then you will have so many clusters that it's hard to deal with. So it's better to put like a lower resolution and group things in bigger groups, and then take that subgroup and recluster that one in higher resolution. But yeah, that's I guess how I would recommend. But yeah, there is different ways of doing the clustering. The k-means was kind of at the beginning, and is again something that was inherited from bulk RNA-seq, but now most of the people use k-nearest neighbor, which connects each cell to its most similar cells. And you can look at this review here about clustering, but this is kind of the default of Seurat at the moment. So that's the easy way to go. And then once you have your clusters and you're happy with the resolution, you can do an annotation. So here now we need to look at what are the cells that we have. So you would find cell markers for each cluster, and then you have to compare these cell markers to reference databases. For example, the Human Cell Atlas, there is also one for mouse or whatever. And there are some packages that would make this easier, where you don't have to actually get the markers and then you don't have to compare it yourself. You just can plug in the data directly, and it will predict which is the most likely cell type that you have based on the reference that you choose. And Singular is one that works quite well, and I'll recommend it. And then there is another way of doing this, which is kind of a recent, has been developed recently, and it's called label transfer. So in this case, you have a reference annotation that is very well annotated. Maybe you have there is a lot of cells, or they've spent a lot of time looking at each marker from each group, from each cluster, and they have this very beautiful annotated reference. And then you have your cells that are from the same tissue. And instead of going and trying to look for all of these markers again and going through the process, what you can do is use this reference annotation and directly look at which cells are the most similar to which of the reference cells. And you can even plot it in the same UMAP that your reference annotation has. The thing is that in order to do this, you need to have a single cell RNA-seq that is very well annotated in the same type of tissue than your data. And one thing that is very important is that all of the cell types from your data should be present on the reference, because if the reference doesn't have a cell type that you expect to see, will not find it. So it has some advantages because it is much faster at identifying cell types. But if you have some new things that you didn't expect to have in the reference, then this is not the way to find it. And maybe it's better to blast your data against more general data sets that have many different types of cell types, such as the Human Cell Atlas, and find that over there. Yeah, but this is the vignette. The integration mapping is the vignette for this label transfer approach in Seurat. Okay, so what I just explained about the clusters and the cell identification is based on discrete analysis. But in biology, not always is black and white, and sometimes you have a data set that is a developmental process, and you have a stem cell and a differentiated cell and a bunch of in-between status. And it might be harder to look at this when you're looking at the clusters individually on how everything connects to each other. And for that, they have developed the trajectory analysis, where you infer what is the order of events that happen based on a developmental pathway. And for trajectory analysis, you can either do unsupervised, where you just take all of the differential genes that you have, and it orders your cells based on those differential genes between what is like, oh, like this cluster is similar to this one, and then that one is more similar to that one, and then like that. So basically on a line or in a branch. Or if you already know what developmental pathway there is in your data set, and there is some very well annotated gene list for that developmental pathway, you can say, okay, I want my cells to be ordered based on the expression of the gene sets that I choose. So that will be a supervised way to generate that trajectory analysis. And one thing here is that this is called, they like to use the word pseudo-time trajectory because it's kind of the process over time. But to actually set the time, so what goes first and what goes after, you need to know either because you have done a temporal study and you know which cells are supposed to be first and which cells are supposed to be later, and then you can say, okay, so this cluster here have my early time point, this cluster here have my late time point, so I know this is the direction. Or based on, I know which cells are stem, which cells are more differentiated because of the markers, and then you can say, okay, so this is a stem, this is differentiated, that's the direction. But the trajectory analysis doesn't give you the early time point unless you do have an early time point or you know what happens first. I don't know if that explained it. It's just my PhD student got confused here. So that's why I mentioned it. Yeah, and then so there is different packages to use trajectory analysis. There is actually quite a lot. So again, I put a review here if you want to look at which ones are the best performing packages, which I think they do also systematic comparison. Monocol 3 is quite widely used, and it has a very easy plugin with Seurat. So it communicates with Seurat. So maybe it's a good way to start, but it's always also good to try two trajectory analysis, at least to see if you get the same type of result. And then once you have your trajectory, you can then validate that using the expression of genes along that developmental branch, and you can use genes that you know should be expressed at a certain time, or look at your variation, the differential genes if you want to actually identify genes that are expressed at a certain time. And then finally, as I said before, that the trajectory analysis doesn't give you the order unless you know which one is the first, the earliest time point. RNA velocity does give you a bit more of a time frame, because RNA velocity adds directionality to your trajectory. And it does this by looking at the ratio between unspliced RNA and spliced RNA, and predicting based on the unspliced RNA what the cell will become later on, which I think is pretty handy and cool. And yeah, there are few packages here. I have no personal experience with it, so I cannot recommend any, but you can have a look. And I think that if you can, this is actually a very powerful tool. Yeah, and then once you have your cells identified based on cell type or based on the trajectory or developmental state, then you can look at what are the proportions of these cells in your different conditions, and look if there is an expansion of a type of cell type in a condition versus another condition. Ideally, if you can have replicates, and look at that happens consistently between a treatment or a control, it would make the analysis more robust. But yeah, that's probably one of the things that you want to do next. Okay, and then finally, the last type of analysis that I'm going to talk about today is the analysis at the gene level. So you might want to do differential expression analysis between your conditions in a certain cluster or in a certain cell type. And something here that you need to, that is important is that gene expression analysis should not be done in corrected data. So if you had to integrate the data, you cannot use that integration for the differential analysis. You need to use just normalized raw data. And there are two types of methods for differential expression. There is the single cell methods, which are developed for single cell RNA-seq, which all of these ones that I mentioned here are methods that are available with the Seurat package using find markers. And MAST is like also a single cell based method that has been recently developed and apparently outperforms the others because it considers the fraction of genes that are detectably expressed in each cell, so it corrects for that. But then you have also pseudo-bulk methods, which are tools that come from bulk RNA-seq, and they have been kind of adapted for single cell, in which you actually group your cells into a bulk, and you group them based on your biological replicates, if you have them. And actually, there's been a really recent paper in Nature Comms that looks at which type of differential analysis works best. And they actually found that the single cell methods have more false discovery, the higher false discovery rate. So you might find genes that are not real, while the pseudo-bulk methods perform better. The only problem is that the pseudo-bulk methods require replicates, and if you don't have them, then they don't actually, if you create pseudo-replicates, they don't behave as well. But yeah, this is something I actually found out, looked at it, this paper this week. So very recent and might deserve a read if you're looking at differential gene expression in single cell. And then once you have your list of genes that are different between your groups, you might want to look at which pathways there are present, in which pathways are these genes enriched. And you usually use these common databases that I brought here. And then there is, again, a bunch of tools, gene set enrichment analysis, which is very similar to the traditional bulk RNA-seq, where you have a group of genes and you look at which pathways are enriched for that group of genes. Or you can look at the gene set variation analysis, in which is kind of, you can do the opposite. You can look at a pathway of interest and look at what is the variation, the variation of the genes that belong to that gene set on your data. So you can kind of ask the question from one way or the other way. Gene set enrichment analysis will be more trying to see if you don't know what you expect, and gene set variation analysis can be if you already have a group of gene sets that you are interested in. And then finally, you can also look at things like cell-to-cell communication inference, where you look at the expression of ligands and receptors in different groups of cells. And if you find a ligand in a group of cells that is highly expressed and a receptor that matches that ligand in another group of cells, you can infer that these two cells might be communicating. And that's what CellPhoneDB does. And then this package was then upgraded with the NicheNet package, in which you also look at the receptor, the cell type that has the receptor, if it also has the downstream signaling that is usually activated by that receptor, kind of suggesting that that receptor is actually engaged to that ligand and is being activated. And I think this is a very powerful tool for single cell analysis. And finally, I just want to mention some of the current challenges that I think a single cell analysis has, and some of the solutions that people are attempting or trying to working on. So single cell data is noisier than bulk, and this would get better by improving the protocols and making the capture very efficient and very consistent between batches. And there's also been a lot of development around cleaning methods that have improved reducing the noise of data. Then we have the batch effect problem, which is that different conditions are captured separately. And then if you have a batch effect and that's the two conditions you're trying to compare, then the batch effect might hide the biological differences that you want to look at. And a way of fixing this problem could be to multiplex your different biological conditions on the same capture. And you can do that by using an antibody or a lipid tag. So the antibody will basically is an antibody that has a little barcode, and then you stain your cells with that antibody in the two conditions separately. And then once you do the capture, you can mix them and do the capture, and then that barcode will be added to your RNA. And you will be able to see which cell it came from, which sample it came from. And in the lipid base, instead of having an antibody, you have a lipid that has a tag, and that lipid binds to them. It inserts itself into the membrane, which helps with, like, you know, the antibody might be more efficient in one cell type versus the other, and that might create problems. And the lipid should bind in all of the cells the same, because all cells have a lipid membrane. And then the integration of different single cell modalities is again a problem. So if you have different modalities and different cells, it's going to be very hard to compare those two data sets. And I hope that there will be more accessible, high throughput, multimodal technologies that will be able to help with that. Or at least you could maybe have a data set that has a multimodal. So you have in the same cell two modalities and do like less cells with that. And then you can compare that one data set to both RNA-seq and the other modality that you want. But like that, you kind of have created now an anchor in which you have this information in the same cell in your same system. And then some general tips is, if you can, try to consult with someone that has experience doing bioinformatic analysis on single cell. If you can at the beginning, would be great. But if not, once you have done the analysis, it's also good to just talk about what you have done with someone that has experience. Someone checks your code and make sure that everything is correct. Even expert bioinformaticians should always have someone else that double checks their code. Then, you know, this is very much common sense, but stick to the frequently used methods and tools in the field, because the reviewers will be more willing to accept that that is correct. And then if you can validate your results with a complementary tool, or do orthogonal validation of your findings in single-cell RNA-seq. And with that, I think I'm actually almost 1 hour up. But if you have any questions, let me know. And if not, I think my email should be somewhere, but if not, I'm happy to share it now in the chat. And if anyone has a question, can also email me that. Oh, I just saw that there is a question in the chat. For differential expression, you mentioned that it should be done in uncorrected data. If you try to compare clusters in Seurat using differential gene expression, can you use MAST? Thank you. Yes, you should be able to use MAST. And you should be, so you can compare any cell that you want doing with any other cell that you want using differential gene expression. And if they are in different clusters, then you will probably find differences because that's why they are in different clusters. And you can use any of the methods that you have. It's only a matter of that basically every time you correct the data, at least in Seurat, you create a new table or a new essay with new numbers that have been corrected by that. But you can always go back to the first raw counts or normalized counts, and then you can use that table instead for your differential analysis. What makes single cell noisier than bulk? So basically bulk, so with single cell, when you capture your cell in a droplet and you have, this is droplet base, you have your bead, I mean, even in the plate, you basically have your bead with a bunch of barcodes that are coming out, and then things have to bind to that barcode depending on the availability of barcodes that there is, and some genes will attach to that and some genes will not. And that a gene didn't attach to the barcode bead doesn't mean that it was not there. So you only get the highly expressed genes because they are more likely to be attached to the barcode, and you still are playing with a level of randomness from which genes are going to be detected and which genes are not. And then also, while you have the capture, there might be cells that break down on the process, and there is some ambient RNA on your sample preparation that can get encapsulated together with some of your cells. So if your sample preparation is very clean and you don't have a lot of dead cells and debris, then it should have a better data at the end. But if your sample preparation is not as good, then you're going to have ambient RNA that gets captured and creates noise. I hope that answered the question. Okay. Okay, people like the reviews, that's good. Okay, so I think if there is no more questions, I think it's 5 on the dot. I'm just gonna leave my email here because I'm not sure if it's part of the program. But if anyone else has any other question, I will be happy to answer that. So please contact me. And yeah, hope that this was useful, and I hope you enjoyed the rest of the conference. Bye.